Welcome to the mini lesson on the Crossbridge cycle. The Crossbridge cycle is part of what we need to understand about muscle structure and function. And of course, structure and function are themes in our class. We're going to talk mostly during this mini lesson about function, but of course, I'm going to refer to structure as well, because remember, another one of the themes in our class is that all specific functions are performed by specific structures. And specifically with regard to the Crossbridge cycle, we're going to be looking at what happens inside a muscle cell. And a large part of what we're going to be talking about is protein filaments that wind up sliding past one another during contraction, which is the generation of force by a muscle. So those two terms, the Crossbridge cycle and sliding filament mechanism, can more or less be used synonymously, and I think it'll become clear as we move through this mini lesson where both of those terms come from. So as I just referred to the fact that structures and functions are inherently related, I want to talk to you a little bit about some structures inside a muscle cell. And if you look inside a muscle cell, a cardiac muscle cell or a skeletal muscle cell, both of which are striated, and in another lesson we'll talk about what striated means, but it essentially results, it's an appearance in the muscle that results from a regular arrangement of protein filaments. And so if you looked closely enough inside one of those muscle cells, you would see a regular arrangement of protein filaments. If you look at the top part of this picture, what you see is some red lines, which are labeled actin. That is a type of protein, sometimes referred to as thin filaments, and some thicker lines that are labeled myosin, and that is a different type of protein filament, and they are a little bit thicker. And you can see that they're arranged in this very regular pattern. Now, if you look at the very center of that picture, you see that the actin filaments, the red thin filaments, tend to form these zigzag shaped lines that are labeled Z lines, and that's what ties the filaments together. And if you look between the two Z lines, you see a pattern of the actin filaments with the myosin filaments in between the gaps. And then if you look to the left or the right, you see the same regular repeating pattern of the actin and the myosin laid right on top of each other. What you're going to see with the myosin filaments is that these are the purple filaments. They're not only thicker, but they have these little projections that are sticking off of the center purple line, kind of hanging out there in the gap between the myosin and the actin. Those projections are sometimes referred to as myosin heads, and those myosin heads actually are going to be the part of the myosin filament that will span the gap and cross between the myosin and the actin and form a bridge, and that is where we get the term crossbridge from. That's going to be very important in the crossbridge cycle, which is going to allow those filaments to slide past one another. Now, if you look at the bottom part of the diagram, what you're going to see is some circles, and those are meant to represent a cross-sectional view of the same filaments, protein filaments, that are represented in the picture above. The picture above is a side view, but you can see that in cross-section, the purple and the red are meant to represent the myosin and the actin, just as in the picture above. And you can see that each myosin is surrounded by actin and each actin is surrounded by myosin. So just wanted to include that to show you a different perspective so that you can understand the spatial relationship between these filaments. From here on out, though, we're going to look at the side view that's represented in the top part of this diagram. So when we think about how the muscle functions in the sense of generating force, which is a function we refer to as contraction, the force is actually the result of the actin and the myosin sliding past one another, Remember, at the very beginning of this mini lesson, I referred to the Crossbridge cycle as the sliding filament mechanism. Now we know that actin and myosin are in fact proteins, and shape is very important to the functioning of a protein, and proteins have a tendency to change shape as they function. So it may be tempting to think that what's happening when a muscle shortens, or when it attempts to shorten, that process known as contraction, whether it actually shortens or not depends on how much force is resisting it. But it's tempting to think that the actin and the myosin fibers are changing shape and that they are getting shorter. But that's not actually what happens. The length of the actin and the myosin does not actually change, but what happens is that the actin and the myosin slide past one another. The sliding, however, is the result of a change in the protein filament. It's just not a change that results in a shortening of the filament. What happens is that the myosin head changes shape, and I'm going to show you exactly how that works here as part of this lesson. The crossbridge, as I mentioned earlier, is actually formed when the myosin head reaches across that gap, spans the gap, just like a bridge would span a gap between, for instance, two banks across a river or something like that. The myosin head reaches across that gap and actually attaches to the actin, forming the bridge, hence the name crossbridge. So if we look even closer in on these protein filaments, this is you see the same two proteins that we saw earlier when we were looking at the sarcomere, the myosin, which is the thicker filament, and the actin, which is the thinner filament. And you can see that myosin head is in the gap between the main part of the myosin filament and the actin. It's ready to reach across to that actin and form a bridge. The thing I want you to notice is that the myosin head is in a very particular formation. The oval is standing straight up. That is the energized position. Remember, I said that the myosin is going to change shape, and that's going to be an important part of the process of generating force. Right now the myosin is in the energized position, and I'm emphasizing the word energized because I want to talk for a minute about energy. Realize, of course, that muscle contraction requires energy. Well, where does that energy come from? We've talked before about how ATP, which is short for adenosine triphosphate, is the main source of energy in cells. Well, what you see here in this diagram is not ATP, but ADP. And when adenosine triphosphate, which has a string of 3 phosphates attached to it, or is part of its molecule actually, when the bond between the second and the third phosphate is broken, you get ADP, which is adenosine diphosphate, and then an individual phosphate group, and some energy is released. Well, it's that energy which actually causes the crossbridge to go into the energized position. So the myosin head actually has an enzyme called ATPase, and that's what actually breaks the third phosphate off of the ATP and results in an ADP and phosphate. And as I just mentioned, it's the energy that's released in that process that causes the myosin head to change shape. And we know how important shape and shape changes are to proteins. That myosin head is now in the cocked or energized position. Okay. So as the name implies, the Crossbridge cycle is actually a cycle, and so I could have started this description at any point, but I just chose the energized crossbridge as the place to begin describing the cycle. So I'm going to show you a sequence of events, and just as a sort of a preview or foreshadowing, by the time I get to the end of the description of this cycle, what you're going to see is that we're right back where we began with this energized crossbridge. So the second step, I've already mentioned that myosin head is going to reach across that gap, bridge that gap, hence the name Crossbridge, and it's going to bind to the actin. We've already seen multiple examples of where proteins bind to one another. I want you to notice that the ADP and phosphate are still attached to the myosin head. But now the myosin is actually bound to the actin. Now that's not a chemical bond, it's a binding. Okay, and the ADP and the phosphate are still attached. The myosin head is still in the energized position. The next step, what we're going to see is that myosin head changing to the unenergized position. So here's what happens next. You can see that that oval, which is representative of the myosin head, has laid down on its side now. And if the tip of that oval was still attached to the actin, it would pull on that actin to the left. And so those two filaments would slide past one another. Now, what causes that shape change in the myosin? Well, we know that proteins tend to change shape when things either bind to them or unbind from them, and that's exactly what's happening. When the ADP and the phosphate unbind, that causes the myosin to change its shape. The myosin head is going to return to the unenergized or uncocked position. And like I say, that happens when the ADP and the phosphate release. The result is, because that myosin head is still attached to the actin, it's going to pull the two filaments past one another. Well, if this process is going to continue, if the cycle is going to continue, we're eventually going to have to get that myosin head to let go of the actin. And that's what happens in the next step. In this case, ATP, with all three phosphates still bonded, binds to that myosin head, and it's that binding that actually causes the myosin to release from the actin. The myosin is still in the unenergized position because the ATP has not been broken down yet, but the ATP binding causes the myosin head to release from the actin. Finally, the ATP is going to be broken down into ADP and phosphate again. And you see that in the process, the energy released causes that myosin head to stand back up in the energized position. So the two filaments have slid past one another, but the myosin head is recocking, and it is essentially going to grab onto that actin just a little bit farther to the right than it did before. And you can imagine that the myosin heads almost act like little oars if you were to think about the way a boat is propelled through the water. You reach with the oar, you pull, and then you lift the oar out of the water and reach a little bit farther forward and pull again. And over time, it takes many strokes, but over time that can cause the boat to move a good distance. And so by the myosin head releasing, cocking, and reaching onto the actin a little bit farther to the right from where it bound before, it actually will essentially row those two filaments past each other. So the filaments slide past each other, hence the name sliding filament mechanism. And that's what actually generates the force. Now, you may be thinking to yourself, okay, well, what initiates this process? And that's a great question to ask, and we'll address it in another mini lesson. But just as a preview, there are some other proteins that are involved. They are wrapped around the actin filament, and those proteins either block this process or they move out of the way and allow that process to continue. When they block the process, the muscle is relaxed. When they move out of the way and allow the cross bridge to cycle, then that's when the muscle is contracting, and that will be the topic of another mini lesson.